Hello dear students, welcome to the another video tutorial of the mammalian histology. And before these lectures we have seen the different type of tissues, the epithelial tissues, and second uh, most common type known as the connective tissue. And in that connective tissue we have started the various elaborations of this connective tissue, and we have seen the connective tissue proper as well as the skeletal connective tissue. In the skeletal connective tissue we have seen the cartilages and the bone and blood, these three type of the connective tissues. And the fourth common the tissue is the muscular tissue. So this will be the fourth type subtype from these chapter second tissues. So in these muscular tissues we are going to focus on the definition review of what are these muscle tissues and what are these muscular type. In that we are going to see the skeletal or the striped muscles. So the muscular tissues are those tissues, they are attached to the bones, or they occur the visceral organs, or they may occur an organs which are made up from the particular type of the single muscle cells that are nothing but the cardiac muscles. And in today's lecture we are going to focus on the skeletal voluntary or the striped muscles. These muscle known by the three names, that are the skeletal muscles, or the second name is the voluntary muscles, or it may named as a striped muscles. So these muscles fibers or these muscles are attached to the bones and move the bones. Okay, what do you mean? They are providing the extra strength to our skeletal. That's why they are going to known as a skeletal muscles. And these skeletal or striped muscles, okay, so these skeletal or striped muscle are the smallest independent unit of a striped muscle, are called as the fibrils or the fiber muscles. So these fiber muscle cells uh, are multinucleated, they are cylindrical and prismatic in the structure. So their length varies from 1 to the 40 millimeter, and their diameter varies from the 10 to the 100 microns. The fibers are grouped together into bundles called as the fasciculi, or the bundles of these fascicules. And each muscle fiber or cell is enclosed by a membrane named as the sarcolemma. So sarcolemma will be the membrane of these muscles, like the each cell having the its outermost membrane known as the cell membrane. So its thickness of this sarcolemma is less than the 100 angstrom, and inside the sarcolemma there are the nuclei and the myofibrils. So the muscle fibers has several hundred nuclei, and which are flattened and oval shaped, and sometimes they are elongated. So these myofibrils, uh, when the skeletal muscles examine under the microscope, these myofibrils shows an alternating light and a dark bands. So these alternating light and dark bands, or these striations, or they look like strips, hence they known as stripped muscles. And these bands are due to the presence of the myofibrils in it. So the various number of the myofibrils are enclosed or the compact to known and they are all known as the sarcolemma, and these sarcolemma combine to form muscles. Okay, so the muscle contains the tissue found in the animals and the function of which is to produce motion. So this muscle is producing the motion and they are providing a specific type of a structure to our body, hence they are known as the skeletal muscles. And these muscles powers the movement of the multicellular animals and maintains the posture of that particular animal. This muscle is a soft tissue, or they may have an hard, feel like a hard, but they have an elasticity property, and this soft tissue that is highly specialized for the production of the tension, which results in the generation of the force, and that force leads to ultimate motion of our skeleton. Okay, and these muscle cells or myocytes contains the myofibrils, which comprises of actin and the myosin filaments, which slide past each other and producing tension that the changes in the shape of these the myocytes. So myocytes are the cells, and they are providing such a tension that this cell can change their shape. Okay, and numerous myocytes make up the muscle tissue, that the multiple cells, we know that the cell if get aggregated then they are making tissue. So the myocytes are the cell of the muscles and they are going to aggregate and combine a structure known as the muscle tissue, and they control the production of tension in these cells, can generate the significant force that is helpful for the motion or the movement of the body. So these muscle consist of long cylinder cells that are the fibers, and each of a bundle of a fiber or fibrils. So these fine fibrils are within each fibril there are relatively thick filament of the protein known as a myosin and thin ones of actin, which is also a particular type of protein present in this particular fibril. And when a muscle fiber lengthens or the shortens, that means they are get relaxed or the contract, the filament remain easily consist in the length but slide past each other. They may pass each other in the contraction, or they may get stretched into the relaxation state. And this tension in active muscle is producing some cross bridges of the um some particular type of the protein known as the myosin. Okay, and these the work done by these muscle requires some chemical energy which is derived from the metabolism by the food. So how this energy is generated? By means of generating ATPs in our metabolism, that is maybe the our gluconeogenesis or the glucogenesis, and these particular these cycles of the energy production or the metabolism, they are going to release the ATPs, and these ATPs are known very well known to produce the energy, and these energy get utilized by our body. And this chemical energy also get generated by means of this shivering, and shivering is the phenomenon where the skeletal or these our muscles get vibrated and they produce the energy. So shivering is the muscle activity that generates heat warms the body. Similarly, some insects vibrate their wings for a while before flight, or while before the nuptial flights, and heating the muscles to the temperature at which they work the best. Okay, so come to these myofibrils, and since these myofibrils uh, are generally varies from the 1 to the 2 micron in the length, or the in and they have the approximately 300 light and the dark bands in one myofibril, and in each millimeter of myofibril having the 300 to the more than 300 light or the dark bands, and the dark band is called as anisotropic or A band. So we know that the dark band is also known as the A band, and is the actual abbreviation is anisotropic band, and the light band called as isotropic or I band. So under microscope, these isotropic or the I bands allow the light to pass through them easily, hence they are known as the light bands, whereas anisotropic bands or the dark bands, they appear light and the dark shape. Okay, so we are going to see some, these are the some pictures which the changes in the fine structures of the skeletal muscles, so that the you will know about the what are the muscles. So you can see in the picture that is the, these are the one bicep and the tricep muscles from our hand, and they are connected by means of the tendons through the bones. Hence, you know that the connective tissue plays an important role to holding these muscles or the intermediate filaments in these type of the muscles, or the connective tissue in between. They are holding this particular type of muscle, and they are helping these particular structure to adhere this skeleton, and by this bone is connected to the tendon, and tendon is connecting these muscle tissues to the bone, and the the longitudinal section are taken of these muscles, knowing that the various muscle bundles are get compacted in between these muscle tissue, and these muscle bundles are again get sectioned and examined under the uh, best type of the microscope, that they are knowing that these muscle bundles are having the various types of the fascicles or the fascicules, which are having the numerous type of the muscle fibers or the muscle cells including in it. So one muscle fiber or the one muscle cell, or the they are also known as the myofibril, that the one Z line from one Z line to other Z line, that the that particular structure is known as the one myofibril, and this myofibril, after getting enlarged into the microscope, that they are showing the some structures like the Z line into these purple color, the actin filaments into these dotted structures, and myosin filaments that are the dark bands into these red, and they are also known as the gate like structures. So these actin and myosin filaments are uh, doing their activity in this particular type of the sarcomere, and they are contracting or releasing by means of adhering each other, and this particular sarcomere is also going to, we are going to examine that particular sarcomere, So these myofilaments in the striped muscle that is shown in this figure, that the arrangement of the myofilaments in a striated muscle. Okay, so the arrangement of these myofilaments or the cross bridges. Uh, the thin filaments are the actin filaments, which are highlighted by means of this purple color and thin lines, and these zigzag lines, two lines from the one end to the other end. So this line is known as a Z line, and this Z line describes the one sarcomere uh, from this point to that point. Okay, and these cross bridges are the thick filament uh, of the myosin, and these myosin or the actin going to interact with each other, and these in these below these these filament are showing the one sarcomere into the contracted state. So you can see that these in relaxed state in upper picture, that these thin filament or the actin filament are very relaxed, and in the second picture, that is these actin filament are get inverted and get inside to the this thick filament or the myosin, so that the second structure is the contracted structure of this particular myofibril, and the upper structure is the in the relaxed state structure. So as you can see, the muscle is extended in the upper diagram and contracted in the lower one. The thick filament are 1.6 micrometers, that are the 0.0016 millimeter long in the vertebrate striped muscle, but up to 6 micrometers or the 6 microns long in the some arthropods. So these are the state when the muscle get contracted or in the relaxed state. So we are focusing on these myofibril. What are these myofibril are? So these thick filaments are contained by a protein named as the myosin, are the thick myofibroments, and these myofilaments actually the protein named as the myosin molecule, and the size of this thick filament is 100 degree Armstrong, and these thin filament which are comprises by means of actin protein, and these molecules of the actin proteins are 50 Armstrong in the diameter, and their ends are projecting inwards into the eye band. Okay, so as you can see in the picture, that each muscle fiber becomes the shorter and broader when it get contract. As you can see in this picture, that the each muscle fiber get shortened into the contracted state, and each muscle fiber get relaxed, or they get distance in between them is enlarged and into this relaxed state. So each sarcomere which contain actin and myosin also shows contractions. So the particular muscle envelope, that is each myofibril, is surrounded by the network of the endoplasmic reticulum, which is called as a sarcoplasmic reticulum, and each muscle fiber, these are the thin tubes, and get incorporated or the interconnected to each other. So at the region of the eye band, the sacs are wider and are called as the terminal cisternae. So the cisternae means they are look like the same, and they at one end and the other one is the second end, hence they known as the terminal cisternae. And in between two neighboring cisternaes there is a transverse tubule or the T-tube. Okay, and all these T-tubes form the T-system, which are filled with the fluid, and two cisternaes are the T-tubule. Between them are called as the triad. So, so 2 plus 1 is known as the triad, and 2 plus 2 known as the tetrad. So the muscle fibers have mitochondria around the nuclei and between the myofibrils. So these muscle fiber as a connective tissues, they surround each muscle fibers in the sheath of reticular fibrils called as the endomycelium. And the collagen fibers are elastic fibers which forms a sheath around the dozen or more muscle fibers. They called as the fasciculi. As we have seen earlier, the fascicules, the outermost sheath of this connective tissue is called as the epimysium, and these outermost um, cell of this connective tissue known as the epiphyseum. And while the tissue surrounds the fasciculi and dividing muscles by septa is called the perimysium. So the both ends of a muscle has a tendons. As we have seen, the both end of the muscle has a tendon and they are interconnected uh, to one bone to the other bone, and they are help in between them the muscle cells are get uh, connected by means of the skeletal bone, that are the skeletal connective tissues. So these myceliated motor nerve fibers into these sarcomere, they help these muscle to contract or to the state in the relaxed state. So we are going to stop here, and in next lecture we are going to focus on the second type of these muscles, name as the smooth muscle, and the third type name as the cardiac muscles. So thank you guys, we are going to stop here.